Good afternoon to all. How important is to discuss about the global pandemic that has made the world go into a very large crisis? Yes, it's truly the most important topic to head a research. This journey will be on the aspects of knowing about what is coronavirus and the modes of its transmission. On behalf of Group 1, myself Asif and Amna will be doing with the presentation and Ashwini behind the slides.
So
So, the aim of the research
just
is to
just
study.
wait. Wait. Just show your face and present.
Okay.
Just open the video and present. Okay. Yes.
So the aim of the project is to study about SARS-CoV-2 and its transmission. And the objectives behind our research are to study about classification of coronavirus, evolution of SARS-CoV-2, structure of coronavirus, life cycle of coronavirus, risk factors, transmission of the virus, symptoms following infections, and global epidemiology.
Materials and methods required. So in order to make a deal with the objectives to be dealt with, we have gone through various materials, mostly the newspaper journals, which has providing all the information regarding what is happening around the coronavirus, and different journals and various research articles in relation to the studies about coronavirus. So collecting information as the group and reviewing materials collected, after which we have summarized and all the data into the presentation. Review of literature. Coronavirus, which has affected the world in the past 2 decades, SARS-CoV and Middle East Respiratory Syndrome COVID. The SARS, severe acute respiratory syndrome causing COVID, and Middle East Respiratory Syndrome causing COVID viruses has been affecting the world in the past 2 decades. MERS COVID emerged in humans from bats through the intermediate host of camels, and SARS-CoV-2, which has started on 12th December 2019 from a local seafood market in Wuhan, central China, and bats are believed to be the sources. So we would widely cover the emergence of coronavirus from the past 2 decades into a pandemic causing deadly virus of this year.
Classification. SARS-CoV-2 is a potentially lethal type of coronavirus, a subfamily of enveloped, non-segmented, positive sense RNA virus that usually cause mild respiratory tract infections. Here we have the classification chart which shows the nidovirales, which are the order for the coronavirus. Next, we have the Coronaviridae, which shows the family of coronavirus. 4 genus are there, which shows alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. 7 coronavirus cause disease in human called as HCOV or human coronavirus. All HCOVs belong to alpha and beta genus.
Natural reservoirs. Bats are told to be the natural reservoirs for alpha coronavirus and beta coronavirus, while birds for the gamma coronavirus and delta coronavirus. Into the lineages. Alpha coronavirus and lineage A of beta coronavirus, they contribute to about 5 to 15% of all the common cold. Alpha coronavirus, it can cause common cold and GI symptoms like nausea, vomiting, diarrhea, and also bronchiolitis or acute exacerbation of COPD. The lineages are H-COVID 22-9E and H-COVID NL63. Beta coronavirus. It includes 3 lineages and it can cause common cold and GI symptoms only. Lineage A includes H-COVID OC43, which is the most common and most severe among all the strains. And next is H-COVID HKU-I7. The lineage B includes SARS-CoV and SARS-CoV-2 or COVID-19, which we will be discussing. And lineage C includes MERS-CoV.
H-COVID in primates. Primates being the common ancestry in the process of evolution, so we have a quick glance at those pictorial representation. Here, in this pictorial representation, we can see the bat and pangolins are found to be the natural reservoirs. Bat and the bat from the virus, bat and from the virus of from the bat, it undergoes mutation or recombination to form HCOV19. Or in the case of pangolin, either directly or and through an intermediate host, it can also form to an HCOV19. Next, we can in the left part, in the right part, we can have a live infection. That means that ferrets, monkeys, tigers, cats, and dogs can be infected with COVID-19. In the left side, we can see an X, which signifies it cannot be infected, which includes domestic animals like pig, chicken, duck, and lab mice. In the next part, we have a question mark, which signifies that shows the unknown susceptibility to H COVID-19, which includes other domestic animals and dormitory camel.
SARS-CoV-19. So the coronavirus has evolved as a homogeneous population, different from both bad COVID as well as pangolin COVID. So it's important to note that the virus undergoes roughly one genetic change every other week. The virus changes more slowly than others due to the fact that the coronavirus has an internal proofreading mechanism, which is an interesting point to be noted. The changes results in a virus variant that is better adapted to its environment compared to the original. That is, after each and every evolution, the virus gets a rebirth as a new and a stronger virus. The process of selection of successful variant is called viral evolution.
Structure. Considering a research on virus, studying the structure is an important part. So into the structure of coronavirus. Structural proteins, we have a mnemonic called SEMN. Here the S, the top part from the S, we can see an S which shows the spike protein, which has a receptor binding sites that binds to ACE2 receptors. Next, we can have an E envelope protein, and then we have the matrix protein, and then we have HE, which shows the hemagglutinin esterase. Then in the downward part, we can see an N nucleoside, which is labeled here. It is the nucleocapsid.
Role of spike protein. So obviously, structural proteins will have a definite role in the functioning of a virus. What is spike protein? Spike protein is the large type of transmembrane and glycosylated protein. It has two domains, S1 and S2, and it is a trimer. So into the native state, we can find that the spike protein, which is attached to the outer surface of the virus, and the receptor binding site ACE2. It binds to the receptor binding site ACE2 of the host cell surface, and after the intermediate stage, it also has a role in combining and the cell fusion.
Life cycle of SARS-CoV-2. Studying the life cycle is the most important part in any sort of research with dealings with microbes. From this pictorial representation, we can see that the coronavirus here binds to the ACE2 receptor on the cell surface. It enters the cell and causing membrane fusion and viral RNA release, which causes the expression of replicase protein. And then transcription and replication takes place. RNA replication and packaging goes hand in hand. For the packaging, assembly and budding is helped by the proteins formed on translation. Next, on the top part, we can see after packaging, these virus cells ready to be released, and then it released out of the cell surface to infect other cells.
For immune response. So after the encounter of a foreign particle, here which is the coronavirus, every cell will have a defense mechanism. So after the entry of a virus, it can lead to an unbalanced immune cell activity. So it will develop antibodies about after 1 to 3 weeks after the symptoms. It will produce CD4 cells and helper T cells, which will stimulate the B cells to produce antibodies specific to the virus. There are both IgM antibodies and IgG antibodies. So IgM antibodies which are produced first and disappear after a few weeks, while IgG antibodies are produced at the same time or 2 to 3 days later. How does the T cells act? The cytotoxic T cells will recognize the infected cell, leading to an immune attack, finally getting the infected cell killed.
Now let us look at how the B cells act here. B cells act as independent of the T cells. They act as the memory cell which respond rapidly if they came in contact with the same virus again. The B cells release agglutinin neutralizing antibody which prevents the virus to bind with the receptors, which makes the cell prevalent from the virus attack.
About the structural mechanism of the coronavirus. Let's go to the next important part, transmission.
Transmission here, we have a direct or close contact with the infected person. Infected person can be contagious and the virus can spread from them to other people. Virus spread after infected people sneezes, cough on or touch surface or object. Others get infected by touching this contaminated surface, then touching their eyes, noses, or mouth. Aerosol transmission can occur in specific settings, particularly in indoor, crowded, and inadequately ventilated spaces. Next, we can find the three C's, which are very essential in spreading and in prevention of transmission. In spreading, we can see the three C's like crowded places, close contact settings, confined and enclosed spaces. These overlapping of these three increase the risk of transmission, and the prevention of these will help in succeeding from transmission.
How it gets transmitted through the droplets and aerosols. First, about the respiratory droplets, which will be more than a size of 5 to 10 micrometer in diameter. The transmission occurs when a person touches a contaminated surface or gets caught directly in the spray zone of an infected patient. So as larger and heavier the respiratory droplets are, it is likely to fall from the air before evaporating, so they do not typically make it more than 6 feet before landing. In closed rooms, it can persist for about 8 to 14 minutes. Next, about the droplet nuclei or aerosols, which are less than 5 micrometer in diameter. These are small and buoyant particles that will remain airborne for long periods of time before evaporating. So the inhalation of aerosols can cause transmission. Rather than respiratory droplets, this will last for a longer duration of 3 hours and can last even longer. Processes involving aerosol generation are intubation, extubation, NIPPV, suctioning, bronchoscopy, CPR, tracheostomy, manual ventilation, etc.
Safeguards against transmission of air droplets. As the COVID-19 pandemic is causing havoc problems around the globe, prevention of spread is necessary. Use of safety measures include respiratory protection and PPE. Respirators and masks should be used resourcefully with special consideration for healthcare. Cloth face covering, surgical mask, and N95 mask are effective. N95 respirators are commonly used by highly risk individuals and healthcare workers, while normal mask is used for the regular protection. PPE stands for personal protective equipment. It include gowns, surgical mask or respirator, goggles, and gloves. They are used by healthcare workers and people who are highly vulnerable to infection.
Viruses on different surfaces. First, about surface transmission. We have told about how it gets transmitted through respiratory droplets and aerosols. The other modes of transmission are direct contact transmission, especially hand-to-face contact. And other one is the fomite or surface transmission, which includes surfaces like latex, aluminum, or copper, where the virus can last for 8 hours, and cardboard, which can be about 1 day, and countertops, stainless steel or plastic, which can last about 1 to 3 days, and wood or glass, which can last with 5 days, and there can also be a transmission via fecal-oral mode. What are the factors affecting transmission? According to WHO, whether a virus is still replication competent. It is an important factor. Next one is whether the patient has the symptoms like cough, which can spread infectious droplets, and the behavior and environment factors associated with the infected individual.
Risk factors. Older individuals over 60 years and children are most likely to vulnerable to infection. People with underlying medical conditions like cancer and their specified treatment, chronic kidney disease, COPD, immunocompromised patient, people having heart conditions like CRDA, cardiomyopathy, and people with obesity on BMI over 30 kilogram per meter square, people with hypertension, diabetes, asthma, and other pulmonary disorder, and people with pregnancy, pneumonia, anxiety, depression, chance of return labor are all vulnerable and prone to COVID-19.
Symptoms. So as far as people are concerned, we are aware of a disease when they start to show symptoms. So what are the symptoms of COVID-19? The most common symptoms, as you all may know, are fever, dry cough, and tiredness. And less common symptoms like aches and pains, sore throat, diarrhea, conjunctivitis, headache, loss of taste or smell is also produced. Rash on skin or discoloration of fingers and toes are also a less common symptom. And serious symptoms include difficulty in breathing or shortness of breath, chest pain or pressure, and loss of speech or movement, which will need an immediate hospitalization.
Asymptomatic spread. The term asymptomatic refers to people who are infected but never develops any symptoms. From the data, it is clear that 80% of infections are mild or asymptomatic, while 15% is severe and remaining 5% of infection are critical. Epidemiology. Epidemiology is an utmost important procedure in tackling any new infectious disease. It includes the identification of the source of the outbreak, monitoring and tracking the disease, studying the disease, and developing guidelines for prevention of spread.
Analyzing the first graph, the data has been collected from over different affected continents of the world. We can see that the confirmed new cases has a gradual increase all over the world. And the second graph, it is found the confirmed death. The confirmed death all over the world has been varying. That is, it is decreasing and has been increasing in a varying order in different parts of the world. Considering the epidemiological curve of India, here we have terms doubling time. What is doubling time? It is defined as the time it would take for the number of new daily cases to double and is a key variable in understanding the speed of the growth of an outbreak. And halving time. Halving time of an epidemic is defined as the time taken for the number of new daily cases to halve. And confidential interval or CI. It is the range of values we are fairly sure what our true value lies in. So the first graph, considering the epidemic curve of the new cases in India, in a time period of about September to October, there has been a peak in the number of cases. The doubling time is about 14 days and the halving time 47 days, which has a gradual difference. And the epidemiological curve of the confirmed death. Here the doubling time is about 8 days and halving time 107 days. That is a very gradual difference, and we can find the graph of the confirmed deaths has been varying all over. It has increased to a period of September, October, and it is found to be decreasing in the present scenarios.
Geographic distribution of COVID-19. Over the last few months, we found that every country stood astonished with the outbreak of COVID-19. According to WHO, as of the latest sources of 30th November, we have the confirmed cases of the range 62.3 million and confirmed at 1.4 million all over the globe. In the last few weeks, we have seen a spike in the curve with US at 13 million confirmed cases and 263,000 deaths, while in India, it is of the range 9 million confirmed cases and 137,000 deaths.
Sorry. So after an insight into the subject, we have studied that out of the 4 types of coronaviruses, only the alpha and beta coronaviruses are capable of infecting humans, and they are hence called the human coronavirus. SARS-CoV-2 comes under the lineage B of beta coronavirus, the natural reservoir of which is believed to be bats according to the studies. So we have studied that spike protein in the viral envelope plays a significant role as a virulence factor in these viruses. The fact that SARS-CoV-2 has very high human-to-human transmission capacity was also brought into light on retrospecting and introspecting the epidemiological graphs plotted by the WHO.
Conclusion. From a thorough learning of a myriad of researches and studies, we observed that COVID-19 caused by the SARS-CoV-2 is one of a kind pandemic on the century. On the dawn of the pandemic, the world has come to a standstill. But science hasn't yet given up hope and is yearning forward to snatch victory from the jaws of defeat. And this would be made easier if we steer clear of the 3 C's that makes the viral spread challenging. So that's all about the topic. We hope all our professors and fellow colleagues have a clear-cut idea about the presentation. Special thanks are due to our mentor, Dr. Revisa, for helping us in completing the endeavor. With the constant effects of science and technology, we have reached a stage of being close on getting vaccinated. Let's hope for an end to this crisis and for an even stronger, better future. Thank you.